with regard to your case, we have 5 genes that have seen mutations in ATM, CCND1, FGF19, KRAS, and TP53. Now we'll start with each mutation one by one. ATM is a gene seen in the HRR pathway or homozygous repair pathway. So what these do is they repair the DNA. So what happens is there are certain drugs which can block the repair pathway or these cancer. So what happens is these cancer cells are mutated. So because of it, there's a loss of function. If you give these BRCA drugs, they will cause excessive destruction of these cancer cells if you give these. And the drug that is given for this is PARP therapies. So that is the drug that has been mentioned here, namely niraparib, olaparib, rucaparib, and those drugs.
Yeah.
And then after that you have CCND1. CCND1 is associated with the cell cycle. So now in your normal cell cycle, it is controlled. What happens in cancer is it becomes uncontrolled. So one way of stopping the growth of the cancer is by giving these drugs, which are known as CDK inhibitors, which stands for cyclin-dependent kinase inhibitors. So here you have this abemaciclib, palbociclib, ribociclib. These drugs can help in blocking the progression of this cell cycle.
Okay.
Then afterwards you have FGF19, which stands for fibroblast growth factors. So these create the stroma and all, which create an environment for the tumor cells to grow. For that you have various drugs such as lenvatinib, which can help in this. Then afterwards we have KRAS. Now KRAS is part of the signaling pathway. So generally it is KRAS, then you have NRAS, and then you have BRAF. So what happens is any mutation in all of these will cause significant signaling. These signaling will then give rise to a situation where there is a stimulation continuously happening, causing excessive growth and feedback kind of mechanism. So any mutation in them will cause this signaling and cause more tumorogenic environment. So for them, we can try downstream inhibitors such as trametinib, MEK inhibitors. So here, as you can see, so and another thing is you also have a mutation in TP53. Now TP53 is generally associated radiologically and medically with association with chemo resistance and radio resistance, and also
like I'm just making it simplify. TP53 mutated. Normally TP53 is a gene which is helpful for our body for cellular formation and all. But if it is mutated in a patient, then it is very aggressive. Then these cases are normally resistant to radiation and chemotherapy.
Okay.
So if in a patient I know that there is T53 mutated, then either I will give very high dose of radiation and chemo, or I will add different programs so that if it get registered to one type, another type will work.
Okay.
So T53 is always a yardstick, like if it is very present, it means these cases are very aggressive.
Okay.
And we should treat them also very aggressively.
So that's why I got full dose of radiation.
So also TP53 is associated with, it's normally in the body, it has a role of a tumor suppressor. So any damage or any repair, TP53 is what is colloquially known as the guardian of the genome. So if there's a problem in TP53, there's a very high risk that invariably you may become more susceptible to cancer. And if there's a TP53 mutation in your cancer, is generally associated with what is known as a poor prognosis and requires a little bit more aggressive management upfront.
Okay. But in my case, the tumor load density is less. That's why these drugs are not effective.
I'm not sure about the tumor load density.
It's given here.
No.
Tumor burden.
Tumor burden.
So this TMB and all is just for the tumor, sir. But then that is, we are looking only at that one point. Whether in the entire body, how much of the disease is there, that also plays a role.
So when we went ahead with the medication, this intravenous drugs, we actually went with a combination of cisplatin and biomam both. So with this variance, you think that helped, or I think radiation helped more?
No, 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 I'm telling you. Actual treatment is by radiation. These medicines which are given along with radiation, they
Preventive.
increases the efficacy of radiation.
Okay.
Suppose if you give only radiation without these medicines,
Okay.
it will work 85%, 90% max. But to get almost 98, 99% efficacy, you have to add these drugs. So that's why they are radiosensitizer.
And
They are not actually chemotherapy. They are chemotherapy, but we don't give them in the intent of chemotherapy. If I want to give a chemotherapy, that dose will be very high, almost 4 times.
Yeah.
Here I am dividing it weekly, no, so the very mild dose.
and radiation is concentrated only here where the tumor is.
Actually, radiation is loco-regional.
Yeah.
This means you are treating the tumor and you are treating adjoining areas where there is a risk of involvement by the tumor. Suppose your tumor is supraglottis, where she had.
Yeah.
Now we have to see not only supraglottis, we have to see the lymph nodes which are drained by it.
Correct.
Like this is known as zone cancerization. Suppose due to some food or something cancer has happened, it has affected supraglottis. That does not mean the disease is only limited here. It may be somewhere else around,
Especially the lymph nodes.
lymph nodes and all, which are not seen even in the PET CT.
So for that chemotherapy will help.
No. For that we give radiation in a wide area.
Okay. Okay.
See, her disease was here, but I have treated entire area, both lymph nodes and whole area. So
Okay.
I have literally sterilized the whole area.
Okay.
So that if some cancer is there here and there, it will be destroyed.
Okay.
That's why it is so important that we give full dose.
So that it does not get chance to
Metastasize
metastasize.
or remain there means it doesn't get chance to remain alive
Okay.
or spread somewhere. Because what happens, the cancer does not remain idle. All the time it is spreading locally with lymph nodes, through blood. So we destroy the whole area. So that's why it was a wide area technique.
Okay.
And I have given by different phases. If I give everything at a time, there will be huge reaction. So first I gave something, then I reduced. Again gave, then I reduced. Like that I have gone in 3 phases. Total dose is this, but in 3 phases.
So that BRCA gene is for breast cancer. Is it mutation for breast cancer?
It is more commonly seen in breast cancer, but it's not just specific. It can be seen in ovarian, in men it can be seen in prostate as well.
Okay.
So that is why it is there. But
So what do you think triggers these mutations? Because I'm not taking alcohol, I'm not taking tobacco, I'm not next to firewood. Nobody in my house is a smoker. So I'm not passive smoker also. So what could have
She's in
initiated?
Dharamshala, no pollution.
No pollution also. It's very good area otherwise.
I am not sure exactly because
No,
there are
I'm telling you. See, the cancer happens not by just an instinct, like just due to some effect. Cancer is already decided before your birth.
Okay.
I told no the Knudsen hypothesis. Before your birth, when you were inside the mother's womb, something happened to her, some genetic insult.
Okay.
It may be some virus like German measles and all pregnant mothers get. It may be some radiation from this. It may be some food. It may be something from water even. Chlorine causes cancer. So, due to some factor, when you were inside the womb, your mother was affected, and that has affected your cancer gene. That
Okay.
is the oncogene. So the mutation started that time when you inside the mother's womb. But that insult, genetic, it is known as genetic insult, was not sufficient to cause cancer. So with that mutation, you were born, and you are waiting for the second insult.
Okay.
So suppose at this age, maybe second insult has come. Once second insult comes, how it came we don't know. Once second insult come, your cancer starts. Hello?
Okay.
Yeah.
Nowadays we see this CRISPR technology that will
Yes.
help in reducing all
the
Yeah.
thing is,
Yeah.
actually there's a lot of problems
Yeah,
with
yeah.
CRISPR.
Yeah.
So like I don't know if I can even say, because CRISPR is looking generally at single things. I don't know if it will help in a large. I'm not sure because in also there's so many mutations here. So I don't know if you can use CRISPR. I'm not quite well versed in CRISPR, so I'm not sure whether it would be applicable.
Since we have identified the variants, I think that is what the technology is based on. They will try to rectify these things.
Yeah, but I don't know exactly
Tell me.
what is the normal, I mean, like,
Yeah.
what are they trying to convert it back to? What is their baseline and all of that?
So you have given me a lot of food
Yeah.
to, mulch on.
Yeah.
I can just think about these things and work it out and probably put it something for a layman
So what
to
is the
understand
count
this.
now?
So I've been making videos.
What is the
Last time I
count
came
now?
in, doctor told me something about immunity. So I
Oh.
made a video
Oh.
on that. So I'll also try to do something that is beneficial for layman, because
No, correct. But only
a
problem
lay
is if
person
they're
when
getting
gets the disease is very
chemo,
frightened.
then they drop very fast.
That is
So
my
anyway,
point
let
of
me see
recording
what
all
is the
this.
present count, then I'll start.
Putting it.
Hmm.
Maybe